Thank you all for coming this afternoon for the second in the series of presidential lectures here at OIST. And we have a very special guest today, Christiane Nüsslein-Volhard. And as you'll see from her presentation, not only is she special as a person, she's also special scientifically. And I think in terms of background, a lot of you have already read about her background, but just to mention a few things, I think she actually is a good archetype or model for OIST in terms of her multidisciplinary and cross-disciplinary background. She started out both in biology and biochemistry, decided genetics was necessary, but I think it's fair to say that she's never been held up by the boundaries between different disciplines. She goes where what's necessary to get the experiments done. After coming up through postdoctoral positions, she was a group leader at Embl, Friedrich Miescher, and then became a director, one of the first woman directors in the Max Planck Institutes in developmental biology at Tübingen from 1985 to 2014, if I've got it right. And in addition to her Nobel Prize in Medicine or Physiology, which I think you all know about, she also has many other prizes and honors that she's received along the way. And in addition, she also has served on many important scientific councils, some to do with science itself, some to do with other aspects of the career of science. And in particular, she's been especially important in having a role in improving opportunities for women in science. And I think she's one of the real leaders in that field in an effective sense. And I think just from almost a personal point of view, I think what's very interesting and characteristic of Christiane science is the ability to think about the question before you start and decide what it is you want to know, and then to very carefully think about, if that's the question, how would you try to get the answer? And I think many of us don't do that. We take a question off the shelf, and we don't think about it, and then we try to answer it with techniques that are on the shelf, and we don't think about it. And I think a characteristic of her work has been to think very deeply, what is the actual question, and then how to go about it, and often that means going against the tenets of the field, the beliefs in the field. And she's never been held up by any presuppositions about science. And I think that's a very important thing and a very important characteristic of her work. I think another thing that is true is the important role of technical skill and expertise. That's not always important, but sometimes it is. And I think that's something else that she's brought to bear, that when you know what the question is and you think about how to do it, often there's no way to actually do that. And you have to yourself come up with technical ways to achieve the experiments and get it done. And then finally, I think as we'll hear today, there's another aspect of science that many of us don't think about that much, which is the aesthetic sphere, the aesthetic sense, context of science. And actually, of course, many of the most interesting or important scientific discoveries or observations are themselves aesthetically beautiful, as well as being scientifically important, and that this is an integrated process, and that it's actually, I think, a kind of blurred line between the aesthetics of art and the aesthetics of science. And so this is something that I very much look forward to hearing about today, and so thank you very much, Christiane, for coming. We look forward to your talk. Thank you.
Okay.
Move to the center.
Move to the center.
Yeah.
It's not very pretty, huh?
Not on the scarf. Yeah, that's great.
Okay. So thank you very much for, well, you haven't invited me. Peter Cruz has invited me, and he's not here. And
Who sent his apologies? I was supposed to.
Doesn't help, really. Anyway, when he asked me to give a presidential lecture, I first thought I would tell you about my recent book. I've written a book with the title of Animal Beauty. And so I gave, can you turn off the front light, please? So I gave this as a title, not all, just the first row. Yeah, better.
That's good.
Thank you. Yeah, that's good. Yeah. And this is dealing at the end with working on color patterns on fishes, which is my present interest and fascination. But then Peter Rose sort of dropped the hint that he would like me more to talk about past achievements and my life and how I made my career and things like that. And so I decided to give you a rather general talk about, what I've done so far as a scientist. But in fact, if you think about it, my old life with Drosophila also concerned beauty. And it concerned beauty in another sense than this type of beauty, which is now my fascination. This is how these beautiful color patterns are made in the life of an animal. And this is actually amazing and difficult to understand mechanistically in terms of biological terms. But one should keep in mind that beauty is not a term which is used as a biological term, which scientists use, because beauty is in the eye of the beholder, and it's a subjective impression which cannot be measured. But what it relates to is biological pattern. How do you get patterns from uniformity? How do complex structures rise during development? And there are other systems where you also have this pattern-forming processes, and which you here is probably most strikingly displayed in this butterfly wing. But the principal question is, how does complexity arise from uniformity in biology? And this was the question I asked myself when I started working with Drosophila after my PhD in about 1975. How do patterns arise in the development of an animal? How does a complex organism, a feeding larva, with a manifold of structures developing inside from an egg, which initially is is very uniform and doesn't contain much structure. It looks just like a bag of cytoplasm. And how does complexity arise? Although, according to the second law of thermodynamics, everything should just go to equal distribution. This problem was, of course, a problem which has been attracted many biologists, and for centuries, of course, one couldn't find a solution to this because embryos were so small and you couldn't really see them without a microscope. And at the end of the 19th century, microscopes had been developed, staining methods had been developed, and people started looking at embryos. And one of the pioneers at this time was Theodor Boveri, who actually did see something in the egg in a sort of structure, and he postulated from this, in Ascidian embryos actually, he postulated that there is something in the egg which decreases or increases from one pole to the next. And actually, this is sort of a model where you said the region with higher concentration of this particular substance would be developing differently from the region with lower concentration of these substances. And this is the first model of a gradient which would create complexity based on different concentrations of a particular substance. But at that time, it was very clear to Boveri also that there were no possibilities to somehow get hold of the molecular basis of this mechanism, because much too little was known about anything at this time, about biochemistry and so on. And what people, however, try to do, they try to identify regions in the egg which would have particular organizing activity. And one person, a pioneer in this respect, was Hans Spemann, who was a graduate student of Theodor Boveri, and he worked with frogs and newts And he discovered a region in the newt embryo which, when transplanted to an ectopic site in another newt embryo, would induce a new axis. And he called this region organizer and postulated that there is something in this region which is sort of influencing the surrounding of the transplanted material to form another embryo. And these organizing substances, these were the ones people were looking for. And then followed years of frustration, because people tried to isolate such substances by doing experiments like cutting and slicing and transplanting and fractionizing and applying again, and in many, many systems, and they essentially failed to isolate anything with organizing activity. In the 60s, people started attacking this problem again with different other organisms. One pioneer here was Louis Wolpert, who worked with Hydra, a very small freshwater polyp. I'll show you a picture in a moment. And he came up with the idea of positional information. And he sort of went further than Boveri with this something decreasing or increasing in the egg. And he postulated that there might be substances, morphogens, which elicit different responses at different concentrations, and in such a way that you have a field, an embryonic field, and you have a gradient of a particular substance, and at high concentrations above a certain threshold, these gray cells would be determined, and in the middle region another group of cells would be determined to do a different structure. And you can see that from one substance you can already get 3 different tissues in this field. This theory, although I think it's really simple and attractive, didn't really meet with much enthusiasm among the biologists. And the reason was that no one had ever seen the molecules which would elicit different responses, and gradients have not been shown to exist. And therefore, people were really hesitant and thought this was just elusive. But the search for morphogens went on in many different organisms and different systems. And this gave time for people, theoreticians, to think more about how you can get complexity from uniformity. And a pioneer in this region, in this area, was Alan Turing, who was the inventor of bioinformatics and computers. And he actually made a theory by which two morphogenetic substances in activator and who would interact with each other create patterns. But in his patterns were not really realizable, and they wouldn't match several criteria which were required. A more sophisticated technique was formulated by Alfred Gierer and Hans Meinhardt, actually at the time when I was a graduate student in Tübingen, next door. And so I followed their work when the Alfred Gierer's group worked on Hydra, which has this very simple head structure and a foot, and they tried to find factors which would induce headness and footness. And but it was for them easier to, as physicists, to think about it than to achieve the isolation of such factors. And so they formulated a theory of biological pattern formation based on autocatalysis and lateral inhibition, and 2 morphogenetic substances, one with a long range inhibiting, and another with a short range which would activate itself. And changing the parameters of these substances and the spatial parameters, you could end up with a variety of different patterns which one can also find in nature. And this was actually I found quite attractive, but again, it was not clear what the substances would be which would elicit these different patterns. And and they made no predictions about the molecular nature about these morphogens, and one really didn't get further in the molecular analysis of these structures. So the question, what is the molecular nature of a morphogen? How can one isolate these substances and show that they really work in a concentration-dependent manner? And I was trained in molecular biology at the time, and I worked on transcription, binding of RNA polymerase to DNA, promoters. And but I learned in microbiology courses and also in genetics that in many enzymes in biochemical pathways have been identified by mutations. So when you make a mutant in yeast or in bacteria which blocks a particular biosynthetic pathway, you stop it and you accumulate the previous. I mean, this is a common method which has been done to explore biochemical pathways. And then, I missed out on that people in yeast had already identified genes involved in cell divisions, like Tim Hunt and Paul Nurse and Lee Hartwell. And I didn't really know that, but in Tübingen there was a group working on DNA replication, and they made mutants and identified all the components in DNA replication by making mutants which failed to replicate. And I thought this is probably the approach to get at morphogens, because if you have a mutant which doesn't make the morphogens, there should be no pattern. And maybe if you have a mutant, then and maybe you could fractionate the product of this particular gene from wild-type embryos and apply it to these mutant embryos, and then use this as an assay for identifying morphogens. So I chose to try the genetic approach pretty boldly as a graduate student and not really knowing any Drosophila geneticists. But I jumped on Drosophila by reading the literature, and this is the only animal which was at that time accessible, because the genetics at least was well developed. Drosophila is the best known genetically, was at that time the best known genetically higher eukaryote, and owing to the work of Thomas Hunt Morgan, who had invented this system in the beginning of the 20th century and unraveled the classical transmission genetics, how genes were organized along chromosomes, how they were transmitted to the next generation. And he was a very important figure in genetics and received the Nobel Prize in 1933. He worked on genes affecting, or mutations affecting, the external appearances of the fly, such as eye color, body color. You see here several mutants, which pretty pictures from Edith Wallace. You see here body color, you see eye color, white eyes, red eyes, eosine eyes, many different eye colors, and also eye shape and bristle form. I mean, he just studied traits which affected the structures of the adult flies. And he came up with two important conclusions. The first was, I mean, this was known at the time, each chromosome is present in duplicate, each gene is there twice. And and in the case of Drosophila, you have 3 pairs of different chromosomes, of chromosomes, and then the X and the Y chromosome. So the number of chromosomes is pretty small, and the genes are aligned linearly on these chromosomes. And what he found, and this is important, and people tend to forget that for each organ, there are many genes that affect it. And there is no such thing like a gene for eye, for antenna, or for leg, or for gut. And each organ requires the activity of many different genes located on different chromosomes, spread throughout the genome. He also pointed out that many genes affected more than one feature, and this is what we observe constantly also in modern analyses. You know that there is almost no gene where you can say it makes only this particular, has only this particular function. Many genes are used over and over again in different contexts, in different pathways. So this is the complexity of the genetics, which is really important to keep in mind, because people have sometimes very odd notions about gene for schizophrenia or gene for kidney or something like that. And this So Drosophila has a short life cycle, 12 days about, and it lays eggs, which are pretty large, and they have three molts until they get to a pupa, and then they develop the fly inside the pupa. So what the egg is producing is not a fly, but it produces a larva, and this is a structure which is much more simple than the fly. And one step, at the time, people had already started doing developmental biology with flies, but they looked at the adult structures mostly, and in very complicated manners, because this is not coming out of the egg. So you have to do sophisticated analyses to see how the fly is made from in later time from imagined discs. So I decided to work on this the earliest step, how to get from an egg to the larva. And the larva is complicated enough, but it's much simpler than a fly. So it has a segmented structure with 3 thoracic segments and 8 abdominal segments, a hind end which is clearly recognizable, and a head end. The head is mostly infolded to make life and hatching easier. And the big question, which I sort of wanted to trying to solve, was what is inside the egg guiding the development of this segmented, complicated larval structure. And actually, I asked people at the time, developmental biologists, how are segments made? How is segmentation occurring? And they said, no clue. And this was sort of good. I mean, there was something you could dig in and find something, because nothing is known. It's much nicer than adding a little piece of a puzzle to an already complete picture. I mean, this should keep in mind. Look for problems which are solvable and complicated enough to be interesting. So essentially what I was going for, morphogens which would be in the egg, guide the development of the larva. At that time, not many people worked on Drosophila embryogenesis, and also the structure of the larva was not well determined, and we worked a little bit on working that out and doing some morphology. But some people had already thought about this approach, and one was Alan Garin, who is a biochemist who joined up with Walter Gehring in Yale, and they tried to, they used a mutant which had what they called maternal effect, where the embryos wouldn't hatch if the females were homozygous mutant for an eye color mutant, deep orange. This is one of these cases where a gene has several functions, namely first determining the eye color and also determining whether the egg would develop. Something was missing in the egg of these females, and when they took cytoplasm from wild-type embryos and transplanted it into these deep orange embryos, they would develop further. They wouldn't get normal, but they would show some sign of development, meaning that one could actually use such an assay for isolating if one had a morphogenetic mutant. This mutant is certainly not morphogenetic, so the next step would be to isolate mutants where, in fact, these embryos, which would develop abnormally, showed signs of a pattern which indicated that they might be affected by a morphogen. So this is the structure of the Drosophila larva when it hatches from the egg, and you see it's quite a sophisticated structure. It has these denticles and hairs which helps you distinguishing within limits the thoracic segments from the abdominal segments. It has a ventral side with these nice denticle bands and some sensory organs, and has a dorsal side with this fine hair pattern which can easily be distinguished. And so another aspect which makes this attractive, an embryo has many organs which are all arranged in the proper arrangement during embryogenesis. But when you look at all these organs, you get very confused, and it's very complicated and difficult. So we concentrated on the cuticle only. The cuticle is shed by the embryo. It's an exoskeleton. It's shed by the larva, and it's sort of a dead structure, an extracellular material from chitin. And when the embryos die, because they are, for example, in lethal mutations, the cuticle can be kept and survived, and it doesn't survive. It's also dead, but you can analyze it, and you can clearly discern the structures by looking at these cuticular specializations. such as the hairs. And actually, when I prepared to do these experiments, there was already one mutant which looked very conspicuously like a morphogenetic mutant, and this was the mutant called bicaudal, and it had been described by Alice Bull, an American biologist working together with Ted Wright, and she, in 1966, described a mutant she called bicaudal, which had a mirror image duplication of the hind end of the larva at the anterior. So this is a very spectacular phenotype and amazing to understand how this arises by a single point mutation. Unfortunately, the mutation was very elusive. Most of the females never laid any bicaudal eggs, and sometimes they laid other types of eggs. Now, my first project when I started working with flies was to analyze this mutant in detail and trying to make something out of it. And this failed. This is the worst mutant I ever worked with. But what I did in the first year with flies, I started to develop methods to screen for more mutants. And in one of these pilot screens, when I screened 100 chromosomes, 100 mutated events, I found one mutant which is really spectacular. And in this case, all the homozygous females produce embryos which look like this, are totally dorsalized, have only dorsal hairs on their cuticle pattern, and miss everything which is coming from the ventral side in a normal embryo. And this was very exciting, and actually these two mutants suggest strongly that the embryo in Drosophila is determined by one gradient along the dorsal-ventral, the anterior-posterior axis, which is disturbed in the case of bicaudal, so where you have mirror image duplications of this gradient. As depicted here, Hans Meinhardt told me, because he was around, and he told me it must be that the posterior, that there is a peak of activity at the posterior pole, and normally this decays towards the anterior. And in the mutant, you have less inhibition along this axis, and therefore you get a new peak at the anterior. And in the case of dorsal, I had intermediate phenotypes which lacked only the ventralmost structures and had shallower gradients, and this could also very well be described by gradients along the dorsal-ventral axis. So at this time, in 1979, we had some evidences for these two axes, dorsal-ventral versus anterior-posterior. But these were dorsal was the only good mutant, so we had to do more systematic mutagenesis screens. And this is when I got a job in Heidelberg, where I joined a lab with, joined forces with Eric Wieschaus, who also, we had a lab together. And yeah, this was a bad picture, but from this time. And Eric was an experienced embryologist and geneticist, and he was the first graduate student of Walter Gehring, and I had met him when we overlapped for 3 months in Basel, and we have kept contact, and then we decided to work together on this problem of systematically looking for mutants affecting morphogenesis in Drosophila. There are two types of mutants. The ones which I have already mentioned, the maternal mutants, are the ones where the mother female fails to produce substances which are placed in the egg during oogenesis, and the males have no effect on the phenotype. And you see that in a reciprocal cross, when the female is homozygous mutant, the eggs lack a substance and develop abnormally. When the female is wild type and the male is mutant, the eggs develop perfectly normally because the female is able to produce this substance. Although in both cases, the genotype of the embryos is the same. So this doesn't matter. It's only the maternal genotype which matters. Another class of mutants is where the embryo's own phenotype matters. And you remember each gene is present twice, and when you do crosses, a quarter of the embryos only will receive the mutant allele of this particular respective mutant set, and only these embryos which fail to develop and show an aberrant phenotype, whereas all the others develop normally. For practical reasons, we screened around and we looked around and we looked for other mutants, and for practical reasons we decided to do this type of screen first because it simply is easier. because it requires one generation less to create these homozygous embryos, where to get these homozygous mutant females, and then another generation is a lot more work. So Eric and I decided to do saturation screens for this type of mutants. Saturation means going through a very large number of lines. Essentially, the screen is done by mutagenizing males. The sperm in these males, you cross them to females, and in the next generation you take a single male which comes from a mutagenized sperm, so it's mutant, heterozygous mutant, and you cross it again and do this couple of incrosses until you get females and males which carry the same mutant allele, and when you incross those, a quarter of the embryos or flies progeny should be homozygous mutant, and either they survive, then you don't have a mutation, which is interesting, or they don't survive, and then you look at the eggs and see whether these quarter of the homozygous embryos show a particular phenotype. One has to screen very many of these lines because to be able to say that, yes, we have at least once or twice hit each gene. So we did, on average, 4 times as many as we did in a saturation screen. We did many crosses to be able to hit each gene on average 4 times, and isolated a large number of mutants. In this case, we not only looked for the segmentation phenotype, but we also picked everything which had an abnormal phenotype which was visible in this cuticle, because there were some very interesting phenotypes which might be of use to other people who were not interested in the same questions as we were. So we isolated a total of 120 mutants in 120 zygotic genes with morphogenetic roles, and many different phenotypes affecting either the anterior-posterior pattern, a third or so, 40 or so, affecting this segmentation along the anterior-posterior axis, or another 40 which affected the subdivision of the dorsoventral axis in several aspects, and then we had another class of genes which was responsible for setting up a proper epidermis, and therefore the cuticle also would be affected in mutants. So this is a survey of the first set of genes. We isolated 46 mutants on the second chromosome, and you see there is a great variety of different aberrations. Some which you see are the segmentation mutants. This one, for example, has duplicated anterior regions of the segment. This one has much fewer segments. Another one also, and then some lack the ventral side, others lack the dorsal side, some have no cuticle whatsoever, others have big holes in the cuticle. Fused segments here, and narrow segments here, undifferentiated cuticle. So this type of phenotypes we analyzed and described. And in our first joint publication, we described the segmentation genes, which got a pretty cover picture in Nature in 1980. And we found that the segmentation genes were pretty striking. We had 3 classes of failures to produce the segmented pattern properly. There was one class where a big chunk, continuous stretch of segments was missing. For example, here in this mutant knirps, only the thorax would develop at the hind end, but the abdomen would be fused into one big patch. Then we had another striking and novel class of mutants, the parallel, which we call the parallel mutants, and there, in this case, pairwise fusion or pairwise of every other segment, a patch of pattern, the pattern was missing. And this is the example paired, which also was on the cover picture. And then we had another class of mutants where each segment was present, but only the one part, and this was duplicated. So these were, we called segment polarity mutants, where you had a mirror image duplication of portion of the segment. And so we had these 3 classes which suggested that segmentation would be segments, the subdivision of the larva, of the egg into segments would be performed in a hierarchical manner. First, large subregions would be determined, and then a double segment periodicity, a bigger periodicity, and then finally the segment periodicity would occur. And this is then a summary of 20 of the anterior-posterior phenotypes where you see these are the gap genes with large chunks missing. Then we had these parallel genes, and then we had these segment polarity genes, also several and other genes in this class where each segment was affected, and then some which had head defects. And this actually should say that we have recently summarized. We had never come to really describe these mutants very, very comprehensively and compare them. And so recently, Eric and I sat together and wrote a big review, which appeared 2 years ago in Annual Review Cell and Developmental Biology, if you want to read up on them. Okay, this was very exciting, and we spread. The most important part of this was that at the end we distributed these genes among the community, meaning that whenever people wanted to work on them, they could work on them, and therefore this field developed very fast, that people worked on them and started cloning the mutants and started getting at the molecular biology of the basis of the mutations and the gene products. And we were not so much involved in this, but we went on to do the second part of the screen, namely the maternal screen, the screen for the maternal mutants. And this actually turned out to be much more difficult because of this extra inbreeding step required. And Eric Wieschaus, together with Trudy Schüppach in Princeton, did screens, and we did in Tübingen did screens for these maternal mutants. And in my lab, we did the screens together with Gerd Jürgens, Catherine Anderson, and Ruth Lehmann. And here you see a picture from this time with Gerd and Ruth. So the results from the maternal screens was quite surprising. We got 40 maternal genes, about 40 maternal genes with morphogenetic roles. And in this case, we got much fewer phenotypes. It turned out that we had groups of genes with very similar, if not identical, phenotype, which you couldn't really distinguish, but they mapped at different positions and complemented each other. And we found 3 such phenotypes, which we suggested where the genes would interact together in a system determining a particular region of the embryo. And we found 3 such systems for the anterior-posterior axis and 1 for the dorsoventral axis. And the 3 systems of the anterior-posterior axis, the prototypes are shown here. This is the wild type. You see the head, the thorax, the abdomen, and the telson. And here is the prototype for the anterior system. This is the gene phenotype of the gene bicoid, which has an abdomen and a duplicated telson, instead replacing head and thorax. And then this is a mutant Oscar, which lacks the abdomen entirely and has just a telson, a thorax, and a head. And then we had another third group, which was curious and novel, a torso which affected only the anteriormost and the posteriormost region of the egg. And so we reckoned that the pattern along the anterior-posterior axis requires 3 independent systems, which would be required for these large regions. In the case of the dorsoventral pattern, we found the dorsal phenotype represented by 11 recessive genes, which all shared the phenotype, and then we had also ventralized phenotypes, the opposite, where we had more ventral structures and no dorsal structures. And this was the first representative was the gene TOL, which later turned out to be very important in, homologs turned out to be very important in innate immunity, but I will not go into this. So I just would like to be focused on, several people in my lab focused on different genes, but one was, I just would like to describe to you, this gene bicoid, which turned out to be particularly interesting. And this was studied by Hans-Georg Fronhoever genetically, and later by other people, Wolfgang Drieber and Thomas Berlet, molecularly. In the case of bicoid, you see here the effect is that the anterior portion of the egg is not developing properly, but only the hind end and the abdomen, and the hind end is duplicated at the anterior. In a way, it is looking a little bit similar to bicaudal, and this is where the name comes from, but it does not display this mirror image duplication, but rather could be described better as a deletion of the anterior portion of the egg. Hans-Georg carried out cytoplasmic transplantations to see whether he found anything which is in the wild-type cytoplasm which could rescue the mutant phenotype. And he did a series of transplantations, which you see here. Donor embryos, in this case 3 copies of bicoid to have more activity, transplanted from anterior of wild-type into the anterior of a bicoid embryo could rescue the phenotype and could restore the formation of anterior structures. When he placed this material from the anterior tip into more posterior regions, such as here in the middle, he could find anterior structures in the middle of the embryo. And when he placed it to the posterior tip here, he also sometimes could find anterior structures at the posterior end. So this suggested that there is an activity localized. Needless to say that when took the bicoid cytoplasm from anywhere else, from the bicoid embryo, nothing would happen. He wouldn't find any activity. So he found an activity localized at the anterior tip of the egg, which organizes the pattern along the entire axis. And when the gene was cloned, it was cloned in Markus Noll's lab, and then the in situ hybridization showed that it's the RNA of the bicoid gene which is localized, sharply localized to the anterior tip of the egg. And this was the transplanted material. And this could also be shown by making the RNA in vitro and transplanting it into bicoid embryos, and you could see that they completely rescued the phenotype. One could also, in this case, by injecting very much RNA, induce a head at the posterior end. Wolfgang Driever provided an antibody and showed that the bicoid protein is distributed in an exponential gradient with a high tip at the anterior pole. And we just had to show that it would activate target genes. And it is easy to discern how different concentrations of the same protein can activate different target genes. Here you see, this is just a cartoon to illustrate that. The idea is that at high concentrations, there is enough protein to activate both target genes with a low affinity and also with a high affinity bicoid. At medium concentrations, only the high affinity gene would be activated and not the low activity gene, low affinity gene, and at the posterior where both would not get enough sufficient protein to be activated. So the idea is that from the sharp localized RNA, the protein would be produced and diffuses along the axis, distribute in an exponential gradient, and then you get responses of genes with varying affinity to the bicoid protein. So we had one of these genes is hunchback, which is one of the gap genes which is expressed in an anterior region with a threshold at about 50% egg length above a certain bicoid concentration, which is reached about here. And we showed that the concentration actually is important by changing the shape of the gradient, placing more gene copies into the mother. And when the mother has just one copy, you get a rather shallow gradient and a very small activated, short range gradient with a very short, small activated region of hunchback. When the steepness of the gradient is raised by 4 copies of bicoid, the threshold is shifted towards posterior regions and much larger regions of the egg express the hunchback gene. And this showed actually that the bicoid protein determines position in the Drosophila embryo in a concentration-dependent manner. So it's, by all criteria, a morphogen. And this was the first demonstration of such a morphogen. In the anterior-posterior axis, there are other signals which are localized. The one is bicoid RNA at the anterior pole. We have also OSCA RNA localized at the posterior pole. This functions slightly differently, but also produces, by and large, a gradient. And also the terminal system, in this case, the localized signal is outside the egg from the follicle cells, but also provides gradients which reach into the egg. And this is how the the egg is made by gradients. So we have a maternal pre-pattern in terms of localized signals, and then these gradients emanate and organize the segmentation pattern. In the dorsal-ventral axis, we have just one gradient, and this turned out to be the the protein of the dorsal gene. This is the one I isolated in my first year in Basel, and in this case, it turns out that the dorsal protein is localized in the nuclei at the ventral side, and outside is not in the nuclei at the dorsal side, and this is how this gradient is formed. Again, a transcription factor. And so you see that the pattern is actually a sequence. The patterning occurs by a sequence of molecular pre-pattern, which increase in complexity starting from a localized signal, a gradient, large regions defined, and then a double segment periodicity until finally you get the single segment periodicity, which here is illustrated nicely by a beautiful picture of Peter Lawrence, where you have two parallel genes which alternatingly are expressed in these stripes, which prefigure the segments which you see in the later in the segmented larva. So pattern built on existing pattern, and there is little evidence of self-organization because each pattern is defined by the previous pattern in a quite controlled manner. And we have no evidence for any gradients which work a la Meinhardt by this self-organization between two substances. The later steps are not only guided by transcription factors, but also by intercellular signaling. So in this case, the morphogens spread in the extracellular tissue, and in a rather similar manner, in a concentration-dependent manner, organize the surrounding tissue. And also, this is a cartoon how you could imagine inductions by two touching tissues. Interestingly, many of the genes which affect the anterior-posterior axis and also the dorsal-ventral axis are members of classical signaling systems, and components of the hedgehog notch toll, Wind, BMP, and EGF as signaling pathways have all been identified among the collection of the genes Eric and I found in the Heidelberg screen. So this is what I wanted to tell you about the Drosophila work. Just many people ask, of course, why is it interesting? I mean, we know now how the fly makes their segments, but is this really so important? And the answer to this question is that many pathways are conserved because all animals are related by common ancestry, which is now clear. It was not clear when we started the screens. We had no clue. We just wanted to know how can we describe one organism in much detail, and how can we understand this fully. And now it turns out that many of the results we obtained were quite useful also for other studies. So they are conserved, and actually for a long time, when people started working in vertebrates in the mouse, the safest way to find something interesting to try to get a mouse homologue to a fly gene, and this usually led to quite interesting results. Many pathways exist also in vertebrates like humans, and many pathways used in pattern formation play important roles in growth or in immunity, and some are also oncogenes. And this is where you which is sort of a demonstration, although we didn't have that in the head of our minds when we started the screen. When you focus on something really interesting and fundamental in biology, usually it ends up, even if you don't want it or it's not guided, I mean, it didn't guide your experiments, it turns out to be important also for other processes and can be used in other contexts. I have already talked too long, but Peter told me that I'm allowed to talk longer, and I wondered whether... And I can stop here and take questions, but maybe I would like to try to tell you a little bit about our recent work. I will talk more about this tomorrow in the big lecture, but maybe I can come back to the title, which is the title essentially for this part now. and the animal beauty part. And you know that after we finished the fly stuff, we really, actually, maybe even before we finished the fly stuff, we were not, when we did not yet know that there was so much homology between vertebrates and invertebrates via their common ancestry, which at this time came as a real surprise, although in retrospect one thinks, why not? I mean, they must have some common ancestors, and if they share genes, then they should have quite a few conserved genes as well. At this time, I was thinking about vertebrates and started to study zebrafish because I figured that in zebrafish is an animal similar to Drosophila, where you can do genetics and you can get at biologically important factors in morphogens via mutations in these genes, encoding them. And therefore, we set out to do large-scale screens for zebrafish embryogenesis. And when you have zebrafish, I mean, there is a big group working here on zebrafish, and I don't know how much you look at the fish, but they are really very pretty. And I think it's very important that they're pretty. And we wondered about why they are pretty. First, why are they pretty? And second, how do they get pretty? And I think this problem of animal beauty is understudied. And at the beginning, everyone says, well, it's just the external of the fish. It's not vital. They survive even if they're not pretty. And so what? It's not very important. But if you look around, then you find that fish patterns, color patterns, have very important biological functions. In the life of these fish, it plays enormous roles how they look. And they're used in intra- and interspecies communication in many ways. I don't want to go into detail, but you see here, for example, the flounder. This is camouflage. They adapt in their patterning to the ground, and therefore are difficult to see. In this case, the fish has a striking coloration which tells the other fish, I'm poisonous, don't eat me. This is called aposematism. And then there are other fish which use the red color as attraction, sexual attraction, and other fish like zebrafish and also these cichlids, where the color pattern probably is used for recognition between the individuals of the same species and not excluding those of different species. And you have many more such functions here. The coral reef fishes, which you probably know from the aquarium, are particularly conspicuously colored, and one wonders, how do you get this pattern? I mean, as a painter, you know how this is done, but in nature, I don't know. Anyway, so when you think about how fish acquire, or animals acquire their color patterns, one has to know that in the evolution, we have these very big branches. I mean, the common ancestor, the bilateria of the insects and the vertebrates, is very long ago, and the building body plan is very different for arthropods and for chordates. And the biggest difference is that in the insects, we have this external cuticle, which I have introduced to you already. This has to be shed in each mold. And in this case, the skin cells, the epidermal cells, produce the cuticle by secretion, and they also produce the color which is placed into the cuticle. So in this case, essentially, it's the epidermal cells which are pigmented. And the pattern is made by and large, where one knows it, it's the well-known signaling pathways such as Wnt and BMP and hedgehog, who help in patterning the insect cuticle. And this is, for example, a case of a butterfly, where you see there is a pre-pattern of wing veins, which tells where the black color should be. So often, all these patterns build on previous pre-patterns. In vertebrates, it's quite different. In vertebrates, you have an internal skeleton and a flexible skin which grows while the animal grows. And in this case, the color is made in particular specialized pigment cells. We have 3 types of pigment cells. In the fish, we have melanophores, xanthophores, or erythrophores, and we have iridophores. And the melanophores absorb all light, the erythrophores and xanthophores absorb the blue light, and the iridophores are shiny and reflective, and they change the colors by when being placed over other pigmented cells. And the pigments are known. In the case, this is melanin, and in this case, it's pyridine-based organic pigments, or carotenoids, which are eaten from the food. And in this case, it's guanine platelets, which produce a structural color in crystalline platelets, which reflect the light. The pigments are contained in organelles because they are poisonous, so they're not freely swimming in the cytoplasm. So briefly, just in mammals, mammalian skin, in the melanocytes are sitting in the basis of the epidermis. The epidermis is multi-layered, and they secrete their black pigment into the epidermal cells. Likewise, in the hair, which is made in a follicle, the melanocytes sit at the base and secrete the pigment into the growing hair. In basal vertebrates, fish, amphibia, reptiles, the situation is more complex and more elaborate. They have 3 different cell types, as I showed you already, and these cell types are layered, so the one cell type sits on top of the other. The basal cell type is reflective iridophores, which shield from the sunlight, and then black melanophores, and then topped by iridophores again, and then on the top is yellow xanthophores. And depending on the shape and the superposition of these cell types, you can create different colors. For example, blue can be created by black cells laid with a layer of silvery cells on top, or blue silvery iridophores on top, and then a thin layer of xanthophores. This is the dark stripe in zebrafish. And golden can be produced by very thick, whitish iridophores topped by orange xanthophores. And this is the way different colors are made. So it's quite sophisticated, and of course, you can create very, very many different colors by changing the shape and the arrangement of these cells. So we work on zebrafish for studying the color pattern, and this is much easier, of course, than tigers, cats, or peacocks, which you might find more interesting, but it's really difficult. This process is difficult, and the reason why we don't know much about it is because it arises only late in development. So it's hard to imagine how in a bird's egg or in a mouse embryo in the belly of a mouse, when the pattern develops, how can you do experiments there? So the fish is, of course, ideal because, as you all know, they develop outside the female organism, and all the development can be studied in the dish and can be interrupted and looked at with microscopes or with the eye. You can perform genetic analysis, meaning identifying components by mutations in them, and this immediately leads to the molecules involved. And then the zebrafish has this very pretty striped pattern, and we have an enormous variation among related species, which is a very, very nice feature. So the pattern you see, blue and golden stripes. The male is less golden than the female. They both are striped, so this is probably kin recognition. The male acquires a golden coloration, mostly in sexual activity, and this is perhaps sexual attraction. But we know not much about the behavior of zebrafish. So this is an exciting finding that related Danio species, which are so closely related that you can form hybrids between them, have quite different patterns. So some have even vertical bars. There's here 2 have vertical bars instead of longitudinal stripes. Others have longitudinal stripes, but also spots, dark spots on light background, light spots on dark background. So it's quite fascinating how in such short evolutionary times you get such divergent patterns. You also see that only the zebrafish has striped fins, whereas the others often have special motifs in the fin which are distinct from the body. So the questions we have trying to answer in the last sort of 5-6 years in my lab is: where do the pigment cells originate? Where do they come from? How do they populate the skin? How does the pattern arise, and which genes are involved in evolution? How do you get from one pattern to the next? And the methods we are using is that we do molecular genetic labeling with fluorescent dyes, which you all know, transgenic animals, and follow them in sophisticated microscopy, in vivo imagining over extended time periods, and we make mutants and clone them and look at the proteins And then we analyze the differences between the Danio species. So this is the pattern in close detail: golden, dark stripes, blue, light stripe, dark stripe. And what is interesting to see is that the xanthophores and the iridophores are present in both the light and the dark stripe, but with quite different shape and density. And the melanophores are only present in the dark stripe, and the exact superposition is required to achieve the proper pigment. And this is already a problem which one has to solve. How do they recognize each other, and how do they acquire the correct shape in the respective region? One sees the shapes better when the cells are labeled with fluorescent dyes, and you see here these stellate, longish xanthophores in the dark, covering the melanophores of the dark stripe, and the iridophores, which are also loose and stellate in the light stripe, in the dark stripe, and more compact and dense in the light stripe. Melanophores are only present in the dark stripe, very big cells which are expanded to fill the space. So the life cycle, again, we have an egg which is shed pretty big. A larva hatches within 4 days. It can swim freely and eat. And then, from then on, it develops in the adult. And at 3 weeks only, the pattern arises. And this is in a process which is called metamorphosis, starting at 3 weeks of development, lasting until 2 months or so. And in this period, the metamorphosis, you get the adult fins, the scales develop, and also the body is covered continuously with pigmented cells. And I show you now a movie. Maybe you have never seen an embryo developing, but this is a quite pretty movie of the zebrafish embryo where you have here cleavage from the single cell. You have synchronous divisions to produce many cells, and then they spread over the egg yolk, and then here the germ develops with the muscles coming up at this side, and then the eye is forming here. Here's the tail, and there the brain, the large brain region. And at this time, there is a new structure which is called the neural crest, which lines the neural tube, the invagenating neural tube at this side, and this is the region where the pigment cells come from. Not only the pigment cells, also many other cells which are vertebrate specific and endow the vertebrates with specialized structures, such as the nervous system, the peripheral nervous system, and all the head structures, head bones, and so on. I don't have time to go on to this, but you see the neural crest is a structure which is developing right and left to the invaginating neural tube, and the cells are highly migratory and they are multipotent, and they start migrating towards the ventral side in streams, segmental streams, starting anteriorly and then progressing posteriorly. And they migrate in 2 routes, outside under the skin and inside between the spinal cord and the musculature and the notochord. And you can see that in a time-lapse movie, these segmental streams of neural crest cells, in this case labeled with GFP, under the control of a neural crest specific promoter, MITFA. And you see these cells as they sort of try to find their way and migrate. The larval pattern is simple. It's just straight rows of cells lining the dorsal, ventral, and lateral aspect of the larva, and some yellow cells on top. Essentially otherwise transparent, and only during metamorphosis the cells making the pattern of the striped pattern in the fish appear, and they pop up from underneath. And the question was, where do they come from? Because the neural crest is long gone by this time. And the answer is that they come from stem cells which are laid down during the migration, to the early migration. Here we have these migrating green cells, and this is the red is the spinal cord, and where in each segment these streams, there is one cell left behind, and this cell develops the dorsal root ganglia, including the stem cells of the pigment cells. And at the beginning of metamorphosis, these cells start dividing and produce progeny, which migrate along the spinal nerves towards the periphery of the fish. and then the cells emerge in the skin and organize into the stripe pattern. When we look at the stripe pattern formation, we see that the iridophores appear first along the horizontal myoseptum and like this region where the dorsal and the ventral musculature are separated. And here they appear in segmental clusters, and the cells divide in the skin as differentiated cells to form a confluent, thick first light stripe, which is about 10 cells wide. And at this time, the melanophores appear in the region where they should form the first dark stripes dorsally and ventrally to this light stripe region. The iridophores, which are here marked with red in the membranes, these iridophores change shape and get stellate and migratory, and you see that better in this bright field picture, where you see these shape changes of the iridophores, which then spread over the next regions. In order to follow the iridophores through development, to follow the pigment cells as they appear in the skin through development, Ajit Singh performed a clonal analysis where he marked individual stem cells using Cre-Lox-induced recombination, in which a stop codon is flopped out of a particular ubiquitously expressed gene, producing the fluorescent label red, and then this cell and all their progeny are labeled in red, and you can then follow them and see them as red clones of red cells in the skin. And one can use this also to follow the pigment cells through development, and as it did that, I mean, the fish swim at this time. They are swimming around, and it's not so easy to do time-lapse movies because you can't fix them for a very long time. So what Ajit did is he just caught them and made pictures every day or every other day in order to have a series of pictures which you could superimpose. And you see here, there's an early clone, 17 days, which this clone has been induced at 4 days of development or so. And the red label indicates the dorsal root ganglion, and this is where the clone originated. And you see the clonal descendants, the clonal progeny are both iridophores and also neurons and glia of the peripheral nervous system. And then you see the clone, it's getting bigger, and here the first dense iridophores appear in these segmental clusters, as I've described, and you also see that nerves are labeled. And this shows us at least that the stem cells are not unipotent, they are multipotent. They can produce, and he showed that in later studies, they can produce all 3 types of pigment cells and also neurons and glia, so they are multipotent. And then he followed the iridophores in this next, and this is now a movie made by superimposition of pictures which have been created every day, and you see that the iridophores divide, spread, and then aggregate again at a certain distance. Maybe we show that again. They divide. Here you see the arrow points to dividing cells. They are in a loose shape here, and when they get to a certain distance, they start aggregating again and get dense. And then in this manner, the light stripes are produced sequentially, starting from the first horizontal myoseptum stripe. The melanophores, actually, in this picture, there are also 2 clonal melanophores which do not divide. So melanophores do not divide in the skin. They just pop up and get bigger. And they pop up from new melanophores pop up from underneath. So the stripes are formed starting from the horizontal myoseptum, iridophores migrating up and down, and then aggregating again. And in this manner you have, which in a process we call patterned aggregation, you have a successive formation of these stripes. And the melanophores fill in the region between these dense aggregates of iridophores. So the question is, how do the stripes arise? And do we have a situation as we had it in the fly case, that we have pre-patterns which suggest where the stripes should form or where the iridophores should aggregate? And this is sort of unlikely because we have already seen that many other fishes have very different patterns, so there cannot be a morphological pre-pattern. But there is a morphological pre-pattern in some sense, and this is the horizontal myoseptum. So we found a mutant which lacks this division line between the dorsal and the ventral myotome. And this is the region where the iridophores first come out and they make the first stripe. And in these mutants, the iridophores cannot come out at this point. And what you see here is that they still form stripes in a meandering way, which are not oriented, and they're arbitrary. In every fish, it's different. And this shows us that there is a pre-pattern in form of the horizontal myoception, but does not do more than just orienting the stripes. And all other events of stripe formation is a self-organizing process. So it does not involve morphogens, no long-range signaling. It's just self-organization between the three cell types. And we can show that by mutants in which one or two of the three cell types is lacking. In here we show mutants, double mutants, where only one cell type is present, and you see that if they are only iridophores, they're spread evenly. If they're only melanophores, they also spread evenly, and xanthophores only also spread evenly. And this means that the cells do not fill in an invisible pre-pattern, but somehow they have to organize themselves by interactions with each other. And when we have only one cell type missing, you get some sort of attempts at stripe formation, but not correct, and we need all three cell types. And this can be neatly illustrated by transplantation experiments. These have been done by Jana Kraus in the lab. This is transparent, a mutant which lacks iridophores and has a poor stripy pattern of just rudimentary melanophore stripes and looks really, really pale. But when you do transplantation experiments by transplanting cells from an embryo which can produce only iridophores in such a mutant embryo, you see that patches can form completely normal stripe pattern, suggesting that here is just the iridophores that were lacking for making proper stripes. And you can also transplant iridophores into embryos which otherwise make only you can transplant xanthophores and melanophores into an embryo which can only make iridophores, and then you can get proper stripy patterns in the regions where these stem cells came in. So this means that the cells need each other. Melanophores require iridophores for proper development and for proper stripe formation. And it means that all three cell types must be present and interacting in complicated manners, which have been sort of sorted out by Hans-Georg Frohnhofer and Jana Kraus in experiments looking at double and triple mutants and postulating long-range and short-range interactions between these particular cell types. The question is now which mechanisms operate in these interactions. And for this, you have to study mutants which do make all three cell types, but still they don't form the proper patterns. And we have here examples of mutants which make broader stripes, broader light stripe, broader dark stripes. Spermidine synthase not 2, this is a potassium channel, Idefix, Asterix, and Obelix. And then we have two spotted mutants which encode, where the genes encode connexins, members of gap junctions, and then a gene Schachbrett with a quite different spotted phenotype, which is encodes a tight junction protein. And when one looks at the molecular genetics of these particular mutants, one finds that they all encode integral membrane proteins which are known to be involved in direct contacts between cells, transmission of electric signals similar to a synapse formation or so. And we suspect that this is the process by which these patterns are made, self-organization by particular molecular interactions via these channels and gap junctions. And it's quite striking that none of the known morphogenetic mutants like delta notch or something, no, notch is involved, but in a very complicated way, like these LTK or other signaling pathways are involved, but just genes which are involved in intercellular communication. And we are now working on this, and this is a new process, a new mechanism how patterns are made, and this is quite exciting. And we just have to, we can only summarize here. We have interactions. We can show that there are interactions between cells of the same type, homotypic interactions which regulate the proliferation of these cells, and we have these heterotypic interactions which adjust the shape to precisely form the pattern. And if one of these connexins, for example, is missing, then the xanthophores here do not acquire the proper shape on top of the iridophores. So this is where we are, and now we just have the last question: which genes cause the differences between the patterns in these different species? And I don't want to tell you now because this takes too long. We have just started investigating this because it's not so easy to breed these beasts, and therefore we are limited to the experiments. But what we do is we create mutants in the other species, which is now easy with the CRISPR-Cas9 system, where you can also do genetics in non-model organisms quite easily, and we create chimeras and by transplanting cells between these species, and then we do genome comparisons because the morphology is pretty similar in these species, and also the lifestyle perhaps. So what may change in them is really maybe restricted in part to the color pattern. And so we think we can explain, when we understand what the how the zebrafish is striped, we probably can explain some of these other fish patterns. Maybe these will still be a mystery, and but we can also learn something about vertebrates. Perhaps this is a mutant tiger. Actually, this is an albino mutant where the brown cells cannot acquire the brown pigment, and this is an allele in a gene which we have identified in fish as an albino gene, so affecting melanin synthesis. But it tells you that there are also two types of stripes. One is affected in the albino gene, one is not affected in the albino gene, and maybe we also here have some sort of interaction between these cells. But the peacock, I don't know, you probably will never understand. And this is the people who have done the work. I would just like to point out that my group now is very small, and the most important person is Uwe Irion, this guy here who is working on these different species and has also worked out the connexin genes. And then Hans-Georg Vonhoever, who you know already from the picoid story. He is now the one who collects all these mutants in fish, keeps them and analyzes them. And then this is the undergraduate, and Anastasia Eskova, who works on a different project, which I didn't have time to tell you about. Thank you for your attention.